This module is on translation termination. A ribosome that moves along a messenger RNA has to know when the translation process is over, and this is accomplished by termination signals on the messenger RNA. So let's take a look. Ribosomes move along the message in the 5' to 3' direction, successively going through cycles of elongation and translating the codons in the messenger RNA into a growing polypeptide, shown here as a red squiggle at the top of a transfer RNA. As the ribosome approaches the 3' end of a message, somewhere just prior to the actual 3' nucleotide of that message, the ribosome will encounter one of 3 stop codons, UAA, UAG, or UGA. This stop codon is UAA. The ribosome, when it moves over that stop codon, will actually stall. It's been demonstrated that a ribosome moving at a healthy clip along a messenger RNA actually stalls for a brief period when it hits the UAA, simply because there is no tRNA with an amino acid that can recognize it. That slowdown is just enough time to allow a release factor, a protein, to enter the A site of the ribosome, and the result of the attachment of the release factor to the complex in the A site via the UAA is to cause GTP to hydrolyze and the whole complex to come apart, releasing the newly synthesized polypeptide and, of course, freeing up the ribosomal subunits and the release factor and the message for further rounds of translation, and of course freeing up also the transfer RNA. So everything comes apart and can be used in new cycles of translation. We talked about structures in the cell called polysomes, which are multiple ribosomes translating a message at the same time, and that's both illustrated here on the left and shown in an electron micrograph on the right. And if you look at the cartoon illustration, you can really see cartooned growing polypeptides longer on ribosomes approaching the 3' end of the message, because more of the protein has actually been translated. And there's one of the other stop codons, UAG, shown in this illustration. So this is a typical eukaryotic polysome in the electron micrograph, as you might see it at very, very high power. So let's talk about a few evolutionary issues. The first one is that the peptidyl transferase we talked about during elongation is a ribozyme. The catalytic activity of transferring the growing polypeptide to the new amino acid in the A site resides exclusively in one of the ribosomal RNAs. And it's very much like the RNA-based self-splicing of introns from the messenger RNAs of organelles like mitochondria and chloroplasts that were mentioned in a prior module. Now a few notes on eukaryotic translation. In case it hasn't been clear, all of what I've been telling you so far about translation is based largely on studies with E. coli, with prokaryotes, with bacteria. So how does eukaryotic translation differ from that of bacteria, or prokaryotes generally? Well, first of all, nearly all proteins that you isolate from a eukaryotic cell don't have methionine at the beginning. Remember that in bacteria, in E. coli, 40% of the protein had a methionine at the front end, at the amino terminus, but eukaryotic proteins, generally speaking, don't. In spite of that, if you create a cell-free protein synthesizing system in a test tube using eukaryotic components and eukaryotic messenger RNAs, the polypeptides that are produced do contain methionine at the end terminus. So clearly, the polypeptides that are synthesized in translation in cells begin with methionine, but then there is a post-translational processing step that removes the methionine so that very few mature polypeptides produced by eukaryotic cells can be shown to have a methionine. So this is interesting. Eukaryotes, in fact, have 2 tRNAs for methionine. One of them is called formal methionine, tRNA F-met. And the other one is tRNA met. And the tRNA met, of course, functions to put methionine anywhere in the middle of a chain of amino acids where it belongs based on the coding in the messenger RNA. And the tRNA sub F does indeed get complexed with a methionine using amino-acyl tRNA synthase appropriate to that reaction, but it never gets formulated. It never actually gets a formal group. Eukaryotes simply lack a formulating enzyme, And they also lack formylase, the enzyme that would, of course, take the formal group off of the methionine after translation was well underway. So what does this mean in terms of evolution? Over time, it was no longer necessary to physically formulate the initiator methionine on its initiator tRNA, because by that time many other components of the process of translation initiation had been well established, and it was no longer required that you physically block the amino terminus. That's the speculation to explain why in eukaryotes selection led to the elimination of the formulating enzyme and the formylase. But again, eukaryotes still have the specific initiator tRNA sub F-met, and the methionine is always the first amino acid in a polypeptide chain. That's kind of a relic of evolution, if you will. And that brings us to an end of the discussion of translation. We've seen all three steps: initiation, elongation, and termination, and some differences and interesting evolutionary issues raised when bacterial and eukaryotic translation are compared.